Now we will discuss about the ARC system. The ARC system is a two-component system and it is also known as aerobic respiratory control. So being a two-component system, this ARC has a histidine kinase and a response regulator. Its histidine kinase is known as ARCB and its response regulator is known as ARCA. This is a diagram where you can see the location of ARCB and ARCA. ARCB is a transmembrane protein and it responds to the changing levels of oxygen. When oxygen concentration is low, the ARCB undergoes autophosphorylation and it acts as the histidine kinase. Its histidine gets phosphorylated. Here you can see that the histidine is phosphorylated to HP, means H is phosphorylated to HP and ATP is converted into ADP. Now this phosphorylated histidine kinase activates the ARCA. ARCA is a diffusible protein. Diffusible protein means it is present in the cytoplasm. And now this phosphorylated ARCA acts as a regulator protein. It acts as the positive regulator for cytochrome D oxidase. This positive regulation means it activates the cytochrome D oxidase. Cytochrome D oxidase has very high affinity for oxygen and thereby it can trap the oxygen even if the oxygen concentration is low. Whereas ARCA, phosphorylated ARCA acts as the negative regulator for several enzymes of the citric acid cycle and the glyoxylate cycle. For example, it inhibits succinate dehydrogenase, it inhibits the alpha ketoglutarate dehydrogenase, it inhibits fumarase A, it also inhibits the isocitrate lyase which is involved in the glyoxylate bypass. It inhibits pyruvate dehydrogenase, it inhibits the lactate dehydrogenase, it inhibits amino acid dehydrogenase, beta hydroxyacyl CoA dehydrogenase, and it also inhibits the cytochrome O oxidase. There is another role of this ARCA also which is involved in the activation of F+ synthesis and other sex factors, but that is out of the scope of this topic. So this ARCA responds to ARC system, particularly the ARC system responds to the changing level of oxygen in the environment. Let us have a look at the NAR system. Now NAR, for simplification I will pronounce it as the NAR system. NAR. So this NAR system has two components. It is also a two-component system and this system is activated by the nitrate and nitrite. So we will have a look at this particular system. Now this is a diagram, the A diagram, that represents the activation of this system by the nitrate. So nitrate activates or nitrate phosphorylates the NAR-X. NAR-X, in presence of nitrate, the NAR-X is phosphorylated. So what is the type of NAR-X then? It is histidine kinase. Okay, so it is the first component, histidine kinase. And this NAR-X transfers that phosphate group to other two components, that is NAR-L and NAR-P. So NAR-L and NAR-P, these are two response regulator. So when NAR-L is phosphorylated, it acts as the activator for nitrate reductase, formate dehydrogenase, nitrate excretion and cytoplasmic nitrate reductase. Whereas it acts as the negative regulator for fumarate reductase, DMSO reductase, and periplasmic nitrite reductase, because nitrate is available for the accepting electrons, so there is no need of other reductase, and therefore the fumarate reductase, DMSO reductase, they are inhibited, or their synthesis is inhibited by this phosphorylated NAR-L. In the B diagram, you can see that it is the nitrite that activates the NAR-Q. NAR-Q is for when nitrite is available, then nitrite acts as the activator of NAR-Q. So NAR-Q is again a type of histidine kinase, and this NAR-Q gets phosphorylated to NAR-QP, and then it activates the response regulator. Those are NAR-L and NAR-P. So NAR-L and NAR-P are the two response regulator in this system, and NAR-Q and NAR-P, these are the two histidine kinase protein in this system. And the role of phosphorylated NAR-L are similar as that in presence of the nitrate. So this is a two component regulatory system. So we have discussed three different systems in detail. The first one is the FNR system, ARC system, and the NAR system. The FNR system differs from the ARC system and this NAR system in being a one component system. FNR is not a two component system, remember this. ARC, whereas ARC and the NAR system, these are the two component system. FNR system responds to the level of oxygen. If oxygen is available, it gets inactivated, and in the absence of oxygen, it gets activated. Whereas in case of ARC system, there are two component. One acts as the histidine kinase, other acts as the response regulator. The ARCB acts as histidine kinase and ARCA acts as the response regulator. Whereas in the NAR system, it is the NAR-P and NAR-Q. NAR-P and NAR-X and NAR-Q acts as the histidine kinase, whereas NAR-P and NAR-L acts as the response regulator. So remember these diagrams, these are very important, and with the help of this diagram you can easily understand the regulation response of the organisms to anaerobic conditions. So what you have to remember is the enzymes which are repressed under aerobic conditions and activated in the anaerobic conditions. So if you remember those enzymes or the proteins, you can easily differentiate between which are the enzymes activated during aerobic conditions and inactivated during aerobic condition, and which enzymes are activated during aerobic conditions and during anaerobic conditions, and which are inhibited during anaerobic conditions. So if you go systematically step by step, for example, what is the meaning of two component system, and what happens when the organism is shifted from aerobic to anaerobic conditions, which are the proteins or enzymes changed or replaced under anaerobic conditions, then you can easily summarize these three systems: ARC system, FNR system, and the NAR system. Okay, so ARC system, it responds to the level of the oxygen, whereas this FNR system also responds to the level of oxygen, whereas the NAR system responds to the level of the nitrate and nitrite. So this is about the response of facultative anaerobes to the levels to the changing environment. I hope you have understood this topic. There are some simple questions along with this particular course, objective questions which you have to submit, and there are some subjective questions which you can submit later on. So for today it is I think it is sufficient. Tomorrow we will see the response to other stress factor. Thank you very much.